everyone. Good morning. It's good to see so many of you out on this cold winter morning. We're happy to have you. I'm Joanne Bundy. I chair the Community Education Council, which sponsors the bucket courses. Our planning committee has been busy over the holidays getting ready for our winter and spring season in 2014. And we're confident that you're really going to enjoy the classes that we have coming up. And I want to remind you to look at the orange sheets, which are the announcements, to refresh your memory, particularly about snow days. If we have bad weather on Tuesday night, Wednesday morning, we will have to cancel the class on Wednesday morning. And if that is necessary to do, then we will have the announcement on KGRN. The library will know about it, and Jim Arns will send you an email about it. So check your emails on Tuesday evenings and Wednesday mornings if you suspect that the class might have to be postponed. If you don't have an email address and we have your phone number, we will call you so that you'll be sure to know. And you'll know that the library is also closed. When that's closed, we can't have class. So that's another thing to check on. So please do that. But then know that the class will still be held. We won't miss out on that class. It'll just be a week later. And that, of course, will interfere with our Open Wednesday, which is actually reserved in case we do have to come over and use that day. But don't worry if we miss an Open Wednesday lecture, because that will be put later. So it'll all get in one way or another. Just stay alert. We are especially happy this morning to have Professor Clark Lindgren back. Our evaluation sheets and your personal comments convinced us that you really wanted to have Clark again after his last course. And even though he is an active faculty member, he has made room to squeeze us in, so we're especially appreciative of that. And this is his first class of the 4-class course on neuroscience in 4 literary forms. Please welcome Professor Clark Lindgren.
Thank you. Can you hear me? Am I loud enough? You're okay. Thanks, Joanne, and the members of the committee who invited me back. It's nice to be here. I don't have anything else to do right now anyway. So I wanted to start off with a couple preliminary comments before I get going and give you sort of my overall view of what I'm doing here, why I think this is worth doing, and why I hope this is worthwhile for you to come. So I see that I have 2 roles. One is to be your neuroscience concierge. Right? So you know what it's like. You go to a new city, you've never been there before, you check into the hotel, you know there's all kinds of good food, a lot of good things to do, but you just don't have a clue where to start. And you really don't want to waste your vacation going to all the second-rate places. So you go to the concierge, and the concierge directs you to the good venues and restaurants and keeps you from wasting your time. So that's really one of my jobs, is to sort of help you see the field of neuroscience so that you can actually then go in and enjoy it yourself. And so along the way, one of the things that I'll be doing is letting you know about certain books that I think are particularly good. And it's a really great time, I think, to be alive. Because there's so much exciting science happening every day, and neuroscience is one of the biggest fields, one of the most rapidly growing fields in science. And there's also a lot of good writers out there, people who are active scientists, so they know their stuff, and then they also are interested in communicating and do a very good job of it. So it's a really great time to learn about neuroscience as it's happening, and so I'll try to let you know about that. I'll start by letting you know about one book that I recommend. If you find the topics that I'll be talking about in today's session, and probably next week's session as well, because I don't think I'll be able to get through everything today. There's a book called The Other Brain, and it's written by Doug Fields, Doug Fields is a scientist at the National Institutes of Health. He's an active researcher. He's also written a lot for Scientific American, and this is his first book, which I think is really quite excellent in being both accurate and then also very accessible and interesting. So that's the first task, is to be your entertaining, your neuroscience entertainment committee. Then the second, slightly more serious task is to let you know about how the field of neuroscience can impact you. And I mean broader than the sort of the obvious categories. And so you know, if you're interested in mental illness or neurological disease, of course, neuroscientists would have something of interest to you. But neuroscience is growing by leaps and bounds, and every year neuroscience seems to be encroaching on other fields and having some important things to say about topics that you might not be aware of, topics that have much more of social impact. And so one of the final sessions I'll be talking about in particular looks at the issue of what does neuroscience have to say about the criminal brain, what makes people criminals, and then knowing that and having some idea about that might give us some strategies for dealing with the problem of criminality in society. How do you deal with it? How do you prevent it from happening in the first place? But when it does happen, what's the best way to deal with people who are caught as criminals? So those are the two overarching goals, and I hope I accomplish that a little bit in the next 4 weeks. All right. So today, so I might need that light down a tad. Yeah. All right. So today, what we're going to do is take a journey. And we're going to take a journey through about 150 years of history of how neuroscientists came to understand a particular type of brain cell. But before we start on that journey, we have to take a quiz. Okay, so here's your pop quiz.
Could you turn the sound down just a bit?
Down, okay. Is it just ringing or something?
No, it's just loud.
It's loud, okay. I could talk quieter too, but that's a little. Is that better? Okay. All right. So here's the quiz. So this brain cell becomes more active in response to sensory input. So when you're sitting there and you're getting different kinds of sensory input, this cell actually gets more active, becomes more physiologically active. It communicates with other brain cells by releasing. Missing a space there. Releasing chemical messengers called transmitters. This particular brain cell, this type of brain cell, regulates the sleep drive, makes you sleepy. When it's transplanted from humans to mice, the mice get smarter. Okay. And it's found in greater number in Albert Einstein's brain compared to an average human brain. In fact, it's the only really significant difference between Albert's brain and those of the rest of us. All right, so you got that? So, is this brain cell a nerve cell, which is also called a neuron? Is it a glial cell? And in case you've never heard of glial cells, they come from the Greek word that means glue. A prison cell or a cell phone? D? All right, I won't leave you leave the suspense too long. So prior to a few years ago, and by a few years ago I really just mean probably 3 or 4 years, and even today probably a lot of neuroscientists weren't quite up on this recent change that's happened in the field, most people would have said a nerve cell. Those are the cells, those are the things that make you smarter, right? Those are the things that respond to sensory input, and you'd expect they would be involved, since they're processing information, they'd be the ones that would be involved in sleep and so forth. And certainly you'd think Einstein had more neurons than the rest of us. He didn't. Okay. Those neuroscientists would have been wrong. Because the right answer is D, the glial cell. That's the cell that actually fits all those criteria. So what I'm going to be doing today is talking about how that happened. To tell you a little bit about glial cells and kind of interesting story that they've played out over the last 150 years. All right, so let's go back to well, not that long ago, but about 150 years ago, when people first started doing what is, whoops, went the wrong way. When people first started studying the nervous system, and particularly started looking at the cellular structures of the nervous system. So beginning in about the mid-19th century, which really isn't that long ago, people were interested in the question, what is the nervous system made of? What is its substance? What is it composed of? Now, by this time, the cell theory was well established. And so I'm assuming you have a rough idea of what a cell is, but just in case you don't, I'll show you a diagram of a cell. So this is a typical eukaryotic cell, the kind of cell that you'd find in an animal or a plant. And we're made up of these. Okay, they are semi-autonomous. They're contained inside a membrane. Within the center of the cell is a nucleus that contains genetic material, and then there's stuff outside in what's called the cytoplasm of the cell that carries out the cell function. Okay, now that's all we're familiar with that today because, of course, we know about things like, you can take stem cells and you can create an entirely new organ or an entirely new person from a single cell. So every cell in your body, even one of your skin cells, has all of the information to recreate your entire body from scratch. If we could program it to do that, and that's a little bit tricky to do, but we're sort of getting close to being able to do that. But the idea in the mid-19th century was that it was known that there were things called cells, and that all life was made up of cells. They were either single-celled organisms, and the entire organism was a cell, or when you got bigger, then you just had more of these cells put together. So your heart is a collection of a bunch of cells, your liver is a collection of cells, and so forth. People had no problem with that. They were quite comfortable, that had been worked out. But when they turned their attention to the brain, it wasn't easy for people to come to that same conclusion about the brain. And there were really two reasons for that, from what I can tell looking back at people who've written about the historical, the time, right around like 1850s or so. The first reason people had difficulty conceiving of the idea that the brain could be made up of individual, somewhat semi-autonomous cells was somewhat philosophical, or just didn't make sense. I mean, the brain is you. The brain is what you use to perceive your world. It's what you use to think. It's what you use to control your behavior. The notion that could be made up of small individual units just didn't make sense to people. It was non-intuitive. Just like, the first astronomers convinced us that the Earth was actually rotating on its axis and the sun wasn't actually moving across this flat land. It was the same kind of thing back then. It just didn't make sense given people's understanding of the brain. The brain was just such a different organ than the liver or the heart, and so the notion of it being made up of individual units didn't make sense. Because how could you go in and cut out a little piece of that and still have a brain? It seemed like it had to be sort of all or none. So that was one pretty strong philosophical reason for people to be resistant to accepting the idea that the brain was also made up of cells like the rest of the body. The other reason was more technical, and that is when anatomists looked at the brain, they couldn't see anything that obviously looked like cells. So when they would take brains out of cadavers or animals and chop them up and look at them under microscopes, they just saw a big mush. They could see fibers moving around, and so people thought maybe the brain was like a big network, a big spider web or something that had a lot of fibers and cables running around. But it just didn't look like it had discrete individual cells in it like these other organs did. And then along came this guy, Camille Golgi, who was an Italian physician, pathologist, and liked to just sort of play around in his kitchen with brain sections. The kind of guy. And he invented a technique for staining brain cells, in particular neurons, and it's called the black reaction, and it's still used today. It's a silver nitrate stain, and it has a really peculiar property of very brightly staining cells so that they actually are sharp, have this really black color. And then a very peculiar feature, and that only random cells are stained. And it's, roughly 1 out of 100 cells pick up this stain and they brightly stain, and then the other ones don't pick up the stain at all. And to this day, this technique is still used. It's not completely understood why that works that way. But it ended up being critical because it allowed people like Golgi to for the first time see images like this. This is from one of his early works, in which now you can start seeing things that look a little bit like cells. They don't look like that nice round cell I showed you earlier that maybe is more typical of cells. They have these stringy processes that stick out at each end. But you can certainly start to get the idea that there may be maybe individual units. Here's another picture of Golgi's work. Ironically, even though Golgi developed a technique that ultimately allowed scientists of his day to accept the fact that the brain was made up of individual cells, he never believed it himself. He held on to this particular theory. Even though he had the technique and it really allowed him to see cells, he just didn't believe it. And you see what you expect to see. You've maybe heard that before. That's something I always tell my students. We're programmed to see what we expect to see. And so when you look in the microscope or in your data, you're going to naturally look and see what you think is going to be there. And I think that was part of the case with him. He was so strongly motivated by those earlier ideas I said about how the brain is this whole structure. It has to be all or none. And so he really stuck with this particular idea, even though he really had the technique that showed the cellular basis of the cells of the brain. It was another guy, Santiago Ramon y Cajal, who used his technique, and he was the one who really solidified that idea. He used the same technique, looked at brain sections. He was known, he was a Spanish physician, pathologist. He was already widely known as an incredible medical artist. He would do medical drawings that were just breathtaking in their beauty. And so he turned that artistic ability to the brain and used this technique, and he made just some remarkable drawings. This is one of his drawings that is showing the cells that are in the cortex of a human. And so this would be the top outer surface of the brain, and this would be going down just a few millimeters. And you can see these cells that are, but for us, obviously they look like cells. They're lined up. They have a particular layer. You can see sort of layers in here that are distinct in terms of how dense the cells are and so forth. Here's another. This is one of my favorites of his. This is the hippocampus. I think this is of a rabbit. And you can start to see the network, how you have, for example, a cell here, and it's sending processes that connect up with other cells, and just these really, really beautiful pictures. And his work really finally convinced people that that neurons, although they're contiguous, and although they come really close to each other, and their processes almost touch, that the nerve cell itself is separate. That they're not actually a one long network. Okay? Cajal and Golgi shared the Nobel Prize in 1906 for their discovery, but they disagreed with each other to the very end about the implementation of their results. So what did they think about glial cells? Well, it turns out that Cajal in particular was really struck by these glial cells. In fact, he almost thought they were more interesting than the neurons. And he, in fact, modified the technique so that he could actually stain these other cells of the brain, these glial cells. And so I think I have, there's a picture here, just beautiful images. And by the way they would do these, the way we would do these today is you would have your microscope set up and you'd stick a camera on it and take a picture. That's the easy way. The way they would do it is they'd look in the microscope and then they would draw it. So you really had to have an artistic ability to really make it both be accurate and then also be reveal what you're looking at. So here, this is an early illustration from Golgi, and notice what he does in this diagram. These cells are neurons, and then these other cells around them are glial cells, and so these ones that are labeled with an A are called astrocytes, and I'll talk about some of the different types of glial cells a little bit later. But notice his images really show, here again is astrocytes, and they're just hanging all over these neurons. And one thing that kind of jumps out at me as I look at these slides is there seem to be more of these glial cells than neurons in his images. And sure enough, that ends up being something that's only been more recently appreciated, that glial cells, in fact, outnumber neurons generally in most brains. But Cajal was really fascinated with glial cells, and he didn't have the techniques to really address these questions, but he speculated in his writings that glial cells might be involved in sleep and wakefulness, and also various executive functions like attention, which is really remarkable. So had Cajal taken our quiz, he probably would have gotten it right 150 years ago, or 100 years ago. So what happened in the meantime? So nevertheless, neurocentrism has dominated the field of neuroscience for the past 100 years. Other than that initial work by Cajal and a little bit by Golgi, who first saw these glial cells, the neuroscience I grew up learning, if you learned any of this in school, what you learned, and what everybody has been learning for about 100 years, is that neurons are the dominant cells of the nervous system. They're the ones that are doing all the heavy lifting, all the heavy work. So to give you a sense of why that is, what I'm going to do is first of all tell you a little bit about these cells, since they probably are only maybe vaguely familiar to you right at this point. So first of all, what are neurons or nerve cells? So like any good nerve cell, or any good cell, I'm sorry, they contain a nucleus, that's what's shown right here. And this structure right here is called the soma, which is basically means body. So there's always a body of the cell which contains most of the organelles and materials that a typical cell would have. And what makes neurons somewhat unique is they have these processes that jut out from that central soma. Generally, people would refer to these processes on one side of the neuron as dendrites. Like it kind of looks like a tree in the winter which doesn't have leaves on it, so you kind of get this arbor-like characteristic to these branches. And then there's also usually one longer processes, which is called an axon. Okay. If you look inside that central part of a neuron, you see all of the same things that you find in any other eukaryotic cell. So for example, this structure here with these little holes in it, or pores, is the nucleus. And that's the part of the neuron that contains all of the genetic information that all of the other cells of the body have. And it uses that genetic information to control or to direct the synthesis of proteins, and that happens out here. So these structures out here, can't see them real well in this diagram, but they're little ribosomes, little factories that make proteins. And Jim has been serving as my ribosome, and he manufactured a protein for us while I was talking. He has talents that he doesn't even know he has. So why are proteins such a big deal? I just wanted to make sure you get this idea. I actually do use this in my class at Grinnell College, in my intro class, to talk about proteins. So proteins are the molecules inside cells that pretty much tell the cell what it's going to be, what shape it's going to have, and what structures and what functions it's going to carry out. And basically, proteins are really simple, almost as simple as this little baby toy. They're a long string of amino acids that are hooked together, kind of like this. And each of these has a different color, which you can imagine would be corresponding to the different amino acids that are used to make these strings that we call proteins. There are 20 different amino acids that are used to make all of the proteins in your body, and there are literally an infinite number, not quite infinite, but more than anybody can really, but I can figure out, ways of putting those 20 amino acids together, which ones are used, how many are used, and then in particular, in a particular order. So the genetic code, what it's coding for is this. It's coding for proteins that have a particular sequence of amino acids that are hooked together. And that's a protein. Now, proteins don't generally stay like this. What they do is they fold up. And they fold up because these amino acids interact with each other in particular ways, and so they maintain a particular conformation. And there's one most favorable conformation that a particular protein will take, a particular shape that it'll take. And that shape is what gives the protein its function. So if it's an enzyme that's catalyzing a chemical reaction, it's this particular shape that allows it to do that. If it's a structural protein, again, ultimately comes down to a linear sequence of amino acids that then gives the protein its shape and then gives the protein its particular functions. I find that mind-boggling that all of the complexity of life, this three-dimensional life, comes down to a two-dimensional structure. Okay, it's a chain. Okay, that's what makes proteins. And so neurons, like all cells, are making lots of proteins, and that is all happening in the cell body. And then there's other organelles, which you've maybe heard of. Neurons have mitochondria, which are the organelles that all eukaryotic cells have, which take the energy in the food that we eat and convert it into a form that the cell can use. And the form that the cell uses is a molecule called ATP. And so the cell needs energy to carry out its functions. It can't use chocolate cake to do that like we can. The chocolate cake has to get eaten, digested, and then in the mitochondria converted into ATP, which the cell can use that form of energy to carry out the different functions. Okay. Now, the most important structure of all cells, but in particular neurons, is one that maybe isn't so obvious, but that's the outer skin that surrounds all of the cells. It's called the cell membrane, and it's basically what defines what's in and what's outside of the cell. It's made up of what's called a phospholipid bilayer, which is a fancy name for kind of a fat sandwich. So the membrane is made up of lipids or fats, and it's in a very thin layer. In fact, it's in two layers, as you can see here. So there's one layer of fat next to another layer, and this actually creates the boundary between the inside and the outside of the cell. And what this does is it makes it so that most molecules can't go across the membrane of a cell. It keeps the cell separate from molecules passing between the inside and the outside. Unless there's a few molecules that can pass across a lipid bilayer. The most notable one is oxygen. So really tiny molecules like oxygen, CO2, they can actually slither right through here and sneak past this fatty interior of the cell membrane, but most molecules can't. They're prevented from going across the membrane. So how do things get across? Well, that's where proteins come into play, because these membranes have within them, within the bilayer, they have these structures that are proteins. So these are made up of these chains of amino acids that form a particular shape, and this particular one that's shown here forms like a chalice. It's sort of like a wine cup maybe, and it's functioning as a channel. And so if you look really closely at this, you can see that this particular protein actually has 5 different subunits that come together, and there's a little channel or a pathway that runs all the way through the membrane from one side to the other. And so that's how things can get across, can get across the cell membrane, is through these protein channels. And now the really cool thing about proteins is because there are so many different ways and different shapes, nature has figured out how to make proteins that function as channels, but can selectively only allow certain molecules to pass through. So it would be just like the door on the library out here, only letting men with beards through. You know, we walk up the door and it would open. Anybody else walked up, it wouldn't open for them. It could spot beard and the gender of the person, and it would only let them through. And there might be another door somewhere else that would allow different types of people to go through. This is an incredible biophysical feat that these things do it, but they do it all the time. And it allows them to really, really selectively control what things can go across the membrane. And they can also change right on the spot. So these proteins can all of a sudden change and open the door, let things in, close the door, and so forth. Spend a whole course talking about these things, so I will stop though shortly. So one of the molecules that can move through these protein channels are ions. And I don't know if you all are real familiar, remember your chemistry well enough to remember what ions are, but basically if you took some table salt and sprinkled it into a glass of water, the table salt will dissolve, and the individual molecules that make up that salt will get carried away into the water. And so we have sodium chloride, which is a little confusing because sodium is spelled Na, which is the chemical symbol for that, which doesn't make any sense except it's the German spelling for sodium, I guess. I'm not even sure if that's true. I don't know why sodium is Na, to be honest. I just made that up. And then there's chloride. And so what they do is they dissolve in the water and they separate from each other. And notice that they have a charge on them. So chloride has a negative charge and sodium has a positive charge. And the easiest way to think about charge and how it works is think of the analogy of a magnet. So you know if you have a magnet with a north and a south pole, you try to push a magnet together, the two north poles will repel each other and the opposites will attract. And it's the same thing for charge. And so positive ions will be attracted to negative ions and they'll be repelled from each other. And so that's a really important physical principle that takes place in biological systems, because there's solutions on both sides of cells that are made up of these ions, and they're floating around both outside the cell and inside the cell. And the remarkable thing, which I can tell you just in one sentence and have to move on, but neurons have the ability, because they have these specific proteins in their membrane that only allow certain ones through, is they can create electricity. Okay, so they can basically separate these charges and then build up electricity, and then they can use that to generate electrical currents that flow in and out of the nerve cells and actually flow in the vicinity of nerve cells. Okay? And so, for example, remember my last bucket course I talked about this technique called electroencephalogram, and you've probably seen that where people put electrodes on your scalp. What that is picking up is the summed electrical signals that are being passed between the nerve cells. There's enough of those nerve cells that are sending these little tiny ions through that it gets picked up on the surface of the scalp and can be recorded as an electrical signal. Okay? 3 more minutes, or no, what do I have? I have a lot of time. It's 7, 8, okay. So, all right, so what that means is that neurons are exciting, and they're easy to observe when they're behaving or when they're not behaving. And the reason for that is because you can take a neuron, and if you put a recording electrode in it that will detect this electricity, okay, just imagine the kind of thing that an electrician brings into your house to stick in your socket to see if your wiring is working. Same kind of thing, you can stick a probe in a cell and you can measure that electricity. And when you do that, neurons just do exciting things. When you inject current, they basically rapidly change their voltage and they fire these what are called spikes. And those spikes or action potentials rapidly move down these processes that I showed you, the axons, and are firing all over the place, all over the brain. And they're relatively easy to record. Now what happens, in addition to having this electrical activity, these electrical spikes, neurons then communicate with other neurons. So even though they're separate, as Cahal showed, they do come in really close proximity. And the way neurons communicate with other neurons is at a structure called a synapse, which is shown here. And this has been cut out so we can see inside the nerve terminal. But here's one nerve cell here, and this is the axon, and an electrical signal is firing down this axon, and when it reaches the end of the axon, it then releases chemicals that are contained within these small balls that are called vesicles, and those get released onto the receiving neuron, or what's called the postsynaptic neuron, and the process begins again. That postsynaptic neuron then gets activated, and it sends a signal to another neuron. And so what you have, to make a really long story very short, is in your brain you have all of these neurons that are connected together. Here's one neuron, and it's forming a synapse with this neuron, and it's forming a synapse with all of these neurons. And you've maybe heard this number, but people have estimated there are about 100 billion neurons in the human brain. Each one of those neurons makes about 1,000 synaptic contacts on average. So that ends up being 1 trillion, right? Yeah, 1 trillion synaptic connections in your brain, which is, astronomical. is a good use of that term. So there this is incredible complexity, and it forms this network. And so each of our brains is made up of this network of neurons that are interconnected at these synapses, and messages are zooming around from one neuron to the next and being passed across these synapses. And so what I wanted to do is show you the following video, which is a fairly
Billions of
typical...
volts light up the sky during a lightning storm. Our human brain models this.
Stop a second. So I just went on the internet and I just pulled off the first video that I saw, that there's thousands of these that you can click on and see how the brain works. And this is pretty typical. I actually think this might have been done by a project. These might have been students who were doing a project. And it's not bad. It's pretty typical and fairly accurate, as of a few years ago, representation of what happens in the brain.
As millions of volts light up the sky during a lightning storm, our human brain models this on a much smaller scale. Electrical energy and chemical impulses are essential for communication throughout the nervous system. The basic unit of that communication is the neuron.
You didn't know if you went in the brain, you'd hear
Neurons communicate between each other. They receive information and transmit information. Approximately 100 billion highly specialized cells can be found in the human brain alone. Their basic structure is the same, consisting of the cell body, dendrites, and the axon. These neurons are essential to life because they make us do what we do and say what we say. The place where all the metabolic activities happen, the synthetic center of the cell, is the cell body. The dendrites receive messages from other cells. The more branches a dendrite has, the more information the neuron can receive.
Axons pass messages away from the cell body to the other end of the cell. The role of the axon is to then convey that message to other neurons, muscles, or glands.
Unfortunately, the inter-body communication system can malfunction, interrupting the flow of information through the body.
I don't know, I think you get the idea. Okay, so that's the, yes, question?
What is the actual size of a neuron cell?
Okay, so the question is, what is the actual size of a neuron? They come in all different sizes, but they range from, some of the smallest neurons are about 10 micrometers, up to maybe 100. I'm trying to think of, so like a hair is about 100 microns, so in diameter, so
How many
40 microns?
100 microns.
But now for the neuron.
And they range, and so the big neurons might be as big as 100 microns. Those would be really huge neurons. Smaller ones could be as small as maybe 10 microns. So small enough that you can't see them without a microscope, but not, not so small that you have to have like an electron microscope or something to see. You have to magnify it about maybe 100 or 1,000 times to really see them. Yeah. Oh, but then the other thing about neurons is what I was describing was the size of the soma, or the cell body. Those processes that come out, those can be extremely long. So you have neurons in your spine that send axons down to your big toe. And that's all one cell. And so the cell body might be one of those bigger ones that is maybe about 100 micrometers. But then the axon could be several feet long. And that actually is one cell that sends its axon all the way down to the muscles and controls them. So those are exceptions. Most of the neurons within the brain don't have that long of a communicating process.
Now, what's a micron and a micrometer?
Oh, I'm sorry, yeah. So those are the same thing. So the question is, what is a micron and a micrometer? So a micron or micrometer is a millionth of a meter. Or if you want a thousandth of a millimeter. So a millimeter, is like this. A micrometer is a thousandth of that. Yeah.
If that or any of the longer axons get damaged, do they
Oh, man,
generate?
good. We'll talk about that a little bit, but so the short answer to that is, and the question is, if one of those long axons gets cut, can they regenerate? And the answer to that is yes and no. Yes, out in the periphery. So if you have an accident and you cut a nerve, you maybe have had this happen to you, you've known somebody who's had a nerve cut because they, accidentally, let their circular saw go crazy. Is that what happened to Charlie Duke? Did he do that one time? That will grow back. If, generally speaking, neurons get cut within the brain itself, they aren't able to grow back. And I'll give you one, why do you think the difference is between what happens in the periphery and the central nervous system?
That's why you're here.
I'm here. I should have to do all the work. Actually, the difference is glia. And so we'll actually talk about that. The glia actually make the difference. But I wanted to just finish before we take our break by pointing out what's missing. Okay, this is a fairly standard, this is what your brain is. It's this collection of neurons, forms this network. They're sending these signals, electrical signals down, they get transmitted to other cells, and there's music playing, it's glorious. That's your brain, right? What's missing? Glial cells. Okay, so after break, I'll shift gears and we'll start telling you about these glial cells.
Thank you for taking your seat. I realize that I neglected to remind you to silence your cell phone. And nobody's cell phone went off for the whole first half of the class. So either you remembered on your own or nobody wanted to talk to anybody in the room this morning. So if you will check your cell phones, we'll turn it back to Professor of Neuroscience at Grinnell College, Professor Clark Lindgren.
I forgot to mention at the beginning of class that there are pens and sheets of paper. Go ahead and write a question down if there's something I say that you have a question, and I'll look at those after class, and maybe I'll be able to introduce some of those questions later on. I enjoy getting those and reading your questions or comments or whatever you want to say on the little pieces of paper. So take advantage of that, and then make sure I get those after class. All right, so might need to have. All right, so turn down the music, come back to reality. So then what are these glial cells? And this is the textbook version. This is still basically what you'll read in a typical neuroscience or biology textbook that you pick up. This is what it's going to tell you about glial cells. So first of all, glial cells come in a variety of different types. There's a type called an astrocyte, because it looks like a star, because it's a central body that has these processes sticking out. A type called an oligodendrocyte, Schwann cells. These both form the wrapping that you've maybe heard about, these wrapped nerve cells. I'll mention that a little bit later. Microglia, which are, besides being tiny, as the name suggests, they're the immune cells of the nervous system. They function the same way white blood cells do in the periphery. They attack invading pathogens and destroy them, gobble them up. And then G2 cells, which are a different kind of glial cell, which I won't say anything about. So there's a variety of cells that are recognized as glial cells. And what do? Well, the first function that you'll read about of glia is they hold neurons in place. And in fact, the word glia comes from the Greek word for glue. So early neuroanatomists got the name of Virchow, who was the first one to describe these cells, thought that they probably function kind of like glue to hold the neurons in place, in their proper place. They supply nutrients and oxygen to neurons, a really important function. I see this as being analogous to being like the cafeteria worker. Okay? They support the development of neurons, and so after neurons get born early in development, they have to find their proper location, and so these glial cells guide them to their proper locations. Kind of like the daycare worker, I don't know. As I mentioned about microglia, they destroy pathogens, remove dead neurons. They're like the garbage collectors. And then probably if you knew anything about glial cells, whether you knew these were called glial cells or not, you probably knew that one of the things they do is they facilitate speedy nerve impulse. And I make that analogous to like a highway crew. So once the oligodendrocytes and the Schwann cells, those are really cool glial cells. And you'll find a picture like this in your textbook. And what this is showing, this is the axon that is sending an electrical signal from the cell body out to the axon, out to the axon terminal. And as you move along these axons, they have a wrapping. And you maybe, it is squeaking here. It's a wrapping, and it's kind of, think of it as like electrical tape, insulation tape that actually provides a physical wrapping of the neurons. And so the electricity that's being generated by these nerve axons doesn't escape as easily out into the surroundings. And so it makes the conduction of an action potential much more efficient and actually much faster. And the way it works, I still find this a totally mind-boggling process. During development, so here this is a cross-section looking through an axon, so take any of your axons. Some point early in development, what happened is a glial cell, either an oligodendrocyte or a Schwann cell, depending on whether it was in the peripheral or the central nervous system, came up to this, it started growing around it. Kind of think of this like a snake, progressively wrapping around you and then squeezing you. So here it started here, it's just wraps about 100 times. And because it's a cell, it has this cell membrane, and the cell membrane is made up principally of fat or a lipid fatty material. It's a really good insulator. So it leaves behind its own membrane, and that's actually, so your wrapping that wraps your axons is actually a live cell that is doing it by wrapping its cell membrane repeatedly around these axons and thereby insulating them. Okay. What you can see, what's being shown here is this wrapping gets interrupted periodically along the axon, and so you can see it stops. So this is one wrapping, and then it stops, and the axon has just its naked membrane here, and that's called a node, and that's actually where the electrical signal is generated, and it jumps from one node to the next. And so when your nerve impulse is propagating down these long axons to your big toe, it's kind of jumping from node to node, and it doesn't really lose any electricity in between because of this really tight insulation that's being created by the myelin. And you've maybe heard of diseases like multiple sclerosis. What happens there is people's immune systems attack this myelin sheath, and it disrupts, it kills these cells in different parts of their nervous system, and it prevents the axons. It doesn't kill the axons, but it prevents the axons from efficiently transmitting information. And so people will have symptoms like, might start with numbness or tingling, and then eventually it becomes a really difficult time coordinating their motor activity or whatever. And it's simply because their axons can't transmit information efficiently and sufficiently rapid to coordinate activity. Okay?
May I ask a
Question?
You know, looking at that, it reminds me of a tree, the way the tree grows.
Oh,
And
so it reminds you of a tree and how a tree grows, because you can see the ring structure in a tree. Yeah, it looks very much like that. It's different in that a tree grows, out like this. And so what happens, if this is the axon, the glial cell comes up and just kind of keeps wrapping around it, but it ends up with that same effect of having these concentric circles that are forming a nice wrapping around the axon. And then, actually, this is a good demo. I never realized that. So if this is the axon, here might be one glial cell that's wrapping it, and then here's another one. And there's a gap between those, and that's actually open to the outside, and so the electrical signals can jump from one gap to the next. And they move much more efficiently.
But that gap is not random, is it? It's
So is
purposeful?
the gap random? It is not random. It's precisely, it's incredible, it's precisely spaced based on the physical characteristics of the axon, so that it's always just the right spacing. For a large neuron, like the one I keep mentioning that goes down to your big toe, the separation is about every 2 millimeters, which is pretty big, right? Considering that the dimension, we're talking about dimensions here, the dimension of this axon, or do it right here, is only a few micrometers, a few millionths of a meter, yet it's a couple millimeters long. That's a pretty long distance relative to how thin it is. That's how far the wrapping goes, and then so it really jumps a pretty big distance from one node to the next.
Are you saying that the impulse, instead of going through, jumps around it from the node to the next one?
I wish I had a better picture here. I realize this doesn't show enough of the process. What it does is, let's see, so it goes to say this is one node and here's another node. What happens is the electrical current runs through the axon and then comes out over here. And it actually does circle around and then come back in here again. So it's kind of running around it like that. That's usually the way it's. But you don't lose, the point is you don't lose electricity where it's insulated, or you don't lose very much. And so that's actually what allows it to be.
What purpose does that serve?
Yeah. What? Is
What
that,
purpose does it serve going around like this? I thought it was going down.
so the question is, what is the purpose of having it circle?
Is it getting up strength to go farther?
So the question is, why is it circulate? I don't have time to really give you a good answer.
Right.
But that's a good question. It does in fact though jump from node to node. But the current itself, there's a difference between these little local currents. They're actually doing this. But then it's also jumping. It's also going to so it's also then getting regenerated here. Okay, so that's really the answer to your question. So it jumps from here to here, and then at this node, the signal gets regenerated because there's effectively a little battery across the membrane of the axons, and actually can then insert more current, and then it goes further down. It goes further down. But one thing that's interesting, and I make this point to my students, there's nothing about an axon that determines the direction that the nerve impulse can go. It can go either way. Once it gets started going one direction, it'll always keep going that direction, but it can always go backwards. It's just, in nature, it always gets stimulated at one end, and so the signal's going the other direction. But there are these really weird medical syndromes where people will unintentionally or accidentally stimulate, like with mechanical force, they might actually stimulate their axons, and you'll get antidromic, or signals will be going the other direction, and there's no reason they can't do that. It's just they don't normally do that in physiological systems. But I have to, you can put that question down if you want me to address it more, but I really don't have time to go into more details about that today, unless it's sort of a...
Well, just a quick answer probably. Alcohol or painkillers, do they interfere with this process? Is that how they work?
So the question is, alcohol?
Yeah, if
Alcohol or painkillers, do they interfere with this process? Yes, they do. Novocaine, which your dentist uses, right? What it does is it blocks one of those examples of a protein channel. Remember I mentioned there are protein channels and they can be very specific to only one ion or another.
Right.
So one of those channels is called a sodium channel because it only lets sodium through. So a sodium ion comes up, it lets it through, potassium, chloride, calcium, none of the other ions will go through. That's the channel that starts off the nerve impulse. And Novocaine blocks those channels. So when the dentist injects Novocaine into your mouth, what she or he is doing is injecting a chemical that's blocking those sodium channels in your sensory neurons, so that all the pain and tissue, or all the damage that is being produced by the drill and whatever else is not, it's still happening. And your sensory receptors are still firing, but the information can't progress into your brain. It can't go along these axons to get to your brain because these sodium channels are blocked. So you really only feel pain in your brain. You don't feel it anywhere else. It's only if the signal gets to your brain do you feel any pain. And so if you can stop it before it gets there, you can. Alcohol doesn't do that. It has more central effects. It makes it so that you just don't care as much. Or you care about other things. So before I go on and tell you sort of bring you up to date over the last few years, I wanted to just point out a few missed clues. So some things that in retrospect we can look back and say, what were people thinking, 20, what was I, okay, I shouldn't even point, what was I thinking 20 years ago? Because I was really pretty clueless about glial cells. I thought that they were the blue-collar workers of the nervous system at best. They were the ones taking care of the important cells that get the timpani playing and all the orchestral music. Glial cells don't do that. They just do their work every day and go home. But there were some clues. We can look back on it and say maybe people, other than Cahal, who really did sort of clue in and thought these glial cells might actually be really, really interesting. So first of all, this is somewhat of a difficult number to arrive at, but glial cells really outnumber neurons by as much as 10 to 1 in certain parts of the brain. Certainly certain parts of the cortex, there seem to be more glial cells than neurons. And that doesn't necessarily mean anything. I mean, you probably do have more support blue-collar workers for white-collar workers in general. But there's certainly, just if you were looking at it from simply a numbers perspective, you might have wondered about, if you were back in 1906 trying to decide what you're going to build your career on, you might have thought, well, maybe I should pick the cells that are more numerous. That might be a better shot at figuring out something interesting, but that wasn't the case. This is even more fascinating. The ratio of glia to neurons increases as you go up the phylogenetic tree to more and more complicated organisms, or organisms that have more complicated brain functioning. So if you look at a nematode, like a little simple worm, the ratio of glia to neurons is much lower than when you go up the evolutionary tree to vertebrates, and then when you get to mammals and so forth. So again, if you're interested in the intellectual power that the brain has, that is clearly something that is changing in a very dramatic way. Might give us a clue that glial cells are perhaps doing something more than just providing support. Glial cells are also the last cells to complete the development of the brain. Okay, and so you guys probably have heard this, but adolescents, like my 16-year-old daughter, her brain is not fully developed yet. The part that hasn't fully developed is in the front part of her brain, and the part of the brain that's not fully developed are her glial cells, and particularly her myelination isn't completed, which is why we don't let 16-year-olds do all the things that we might let somebody who's in their 20s do. They just aren't mature enough. They don't have as much ability to control their impulses and all that. It's just their brain is making them do that, and it's really their glial cells. And so, given that we know that when animals develop, they get smarter and they become more capable, and really all of the neurons are in place by the time a baby is born. There aren't any more neurons that are born. They're all there. It's really the glial cells which are doing most of the changing during development. In retrospect, that might have been a clue that they're doing something more than we thought. And then lastly, this is one of the best ones, and I mentioned this in my quiz. So when people finally took a close look at Einstein's brain, and so it's a fascinating story. When he died, there was a pathologist at Princeton, his name was Harvey, I think it was his name, and he did the autopsy on Einstein's brain, and he took his brain. And there's a lot of historical controversy about whether he had permission to do it. He probably did. He just basically stole Einstein's brain and put it in a jar. He fixed it, he was a pathologist, he knew what he was doing, but he hung on to this thing for 30 years. Apparently he doled out little tiny sections of his brain to some of his colleagues, but for the most part he just hung on to Einstein's brain. And then about 30 years later, so in the 80s, some reporter got wind of this and said, did you guys know that Einstein's brain is still around? And so people started getting interested in this, and one person in particular, a neuroscientist at University of California, Berkeley, by the name of Marion Diamond, she persuaded Harvey, this is not Einstein's brain, by the way, she's holding another brain, teaching a class at Berkeley, but she persuaded Harvey to let her have some significant portions of Einstein's brain, and he relented. So that was really the first time anyone had been able to do a really careful study. And she, of course, there was a lot of, I don't know if you remember this, did any of you remember when this was going on in the 80s? I kind of vaguely remember it, but there was a lot of hoopla because after all, whose brain would you want to look at more closely than Einstein's? And it turns out there was, his brain was pretty unremarkable except for two things, according to her studies. One is that he had more glial cells than there were 11 roughly age-matched controls that she looked at, and then she compared that to Einstein's. And there's been a lot written about this, and there are issues of statistics because she was comparing it to one brain. But generally, the effects seem pretty significant, especially in a part of the brain that's called an association area, and that's the part of the brain where you're putting things together, you're taking sensory information and putting them together and coming up with more complex thought. That was really the part of his brain that had was most strikingly different in terms of the number of glial cells. Turns out he also, a later study that just came out a couple years ago also showed that Einstein had a bigger corpus callosum, which is the band of fibers that connect the right and the left hemisphere. And it's also, these are fibers that are myelinated too, and they basically, so he had a better, his left and right hemispheres communicated probably more efficiently than the rest of us. But finally, Einstein, his brain, this really probably was she published this in 1985, 1986, and that probably was really the thing that sort of pushed people to begin starting to look at glial cells and take a second look at them. However, it was really the invention of a new experimental technique that really changed the field. And it's kind of an interesting thing in terms of the history of science. You think of science as progressing by thinking, people sitting down and thinking really carefully. A lot of the advances, if you go back and look at the history of science, and certainly neuroscience, have come about because of a new technique that let people see something they couldn't see before. And this is a really, really good example of that. There was the development of a technique that allowed neuroscientists for the first time to see something quite remarkable. Okay, so what is that technique? So before I tell you about it, though, I need to tell you about calcium. Okay, which, by the way, calcium is probably going to kill you. I always tell my students this, hoping that they'll pay more attention, but they're so oblivious to death. That's so far off. They're immortal as far as they're concerned. But you guys are probably more interested in this. Myself included. By the way, I heard that my picture was in the obituary section of the Grinnell Register. Somebody told me that. But for the bucket course, it was like right next to somebody's obituary. So somebody thought I had died. And realized that, no. Anyway. So calcium is one of these ions. It's like sodium, like chloride. And this is a little bit of a complicated diagram, but just hang in there a second, and I'll explain what's being shown here. So this is a cartoon showing a cell. So this would be the cell membrane, separating the outside of the cell from the inside of the cell. And if you just look right here, the concentration of calcium inside all of your cells is kept really, 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 really low. Okay? Lower than the concentration of calcium in distilled water. I mean, it's just incredibly low. If you're interested in units, it's less than 50 nanomolar, nano being a billionth of a molar. That might not mean a whole lot to you, but it's really, 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 really kept low. And it does that, and it's true of all of your cells. They keep the calcium concentration really low. And the way they do that is they use these pumps, and notice this word ATP is here. So this is one of the places that energy that I mentioned, that mitochondria take energy from chocolate cakes and converts that into ATP. Then the cell uses that ATP to do work, to basically do things like pump calcium out. So think of this as like a pump in a boat that's constantly pumping the water that's leaking in so that the boat doesn't capsize. So there's a calcium pump. There's another one here that does the same thing. It's pumping calcium out in exchange for sodium. There's another pump that's then pumping sodium out in exchange for potassium. And so these together are helping to keep calcium really, really low. There's also a pump that uses ATP that pumps calcium inside an organelle. This is called an endoplasmic reticulum. And so even though the calcium is still inside the cell, it's not in this main compartment, which is where all this stuff happens, where all the reactions happen. Similarly, there's a a transporter that pumps calcium into the mitochondria, which is another organelle. And again, even though that doesn't remove calcium from the cell, it keeps it really, really low in this extracellular space. It does that despite the fact that there are channels that will allow calcium to come in. So we live dangerously. All of our cells live dangerously. And the problem is that if this calcium gets too high, and it doesn't have to get that much higher than it normally is, it kills you. Okay. And in particular, in neurons, it kills neurons. And so if calcium levels get too high, what happens is the calcium then activates these suicide enzymes that kill the cell. Okay. So, and if you kill brain cells, especially if you kill a whole bunch of them all at once, they don't recover their function, and they basically will cause you to die. My wife is a physician, as some of you may know, and so one of her jobs is to, fill out death certificates and say what the cause of death is. And I tell her, your job would be a lot easier if you just wrote on all the death certificates calcium, because she'd be right, and she won't do that. But so what do I mean by that? Well, okay, so you have a heart attack. Why does a heart attack kill you? Your heart stops working, right? So you're not pumping blood to your brain. So your brain isn't getting nutrients and oxygen. The mitochondria and the brain cells can't make ATP, and everything goes. All these basically finely tuned pumps and mechanisms start to fall apart, and the calcium levels start to rise, and then it triggers these suicide enzymes, and your brain cells die. You have a stroke. I mean, basically, I think I read a book once that said 95% of deaths are caused by calcium, ultimately rising. It's the very final thing that happens. It might not be the event that precipitated that, but the very final thing that happens is the calcium rises and it kills the neurons, and then the neurons are dead, and you no longer have a functioning, living human anymore. Okay? So calcium is really, really deadly. By the way, in case you're curious, the only other way you can die, if you're interested, is to be squashed like a bug. Like that'll kill you as well. Like really severe mechanical damage will also destroy your brain without going through this intermediate process of the suicide enzyme. I'll let calcium kill me. So you might wonder, if calcium is so dangerous, why do we have it? And why do we use it? Well, it's also really, it's also really good. So calcium does all of these things for us. Elevations of calcium inside cells, for example, are responsible for sperm motility and fertilization. So like my mother always told me, I brought you into the world, I can take you out. Calcium brings you into the world, and it's also probably going to take you out. Okay. It triggers contraction of muscle cells. It causes the release of neurotransmitters as well as hormones. So they don't get released all the time. They have to be told when to be released, and that in a brief, that's the key word, a very brief and transient increase in calcium is what triggers the release of neurotransmitters and hormones. Not enough to kill you. It controls synaptic plasticity, which is a fancy word that means synaptic change. And so the changes that happen at synapses are responsible for things like learning and memory. So your ability to change over time and learn things and remember them really comes about because of changes that happen at your synapses, and calcium is controlling that. And there's cellular enzymes, regulations of genes, cell growth. So calcium is used by all different cells for really important functions. And if we didn't have it, we wouldn't be able to do all of those things. But like anything, too much of a good thing can kill you. Oh, by the way, notice on this list, muscle cells contracting, neurotransmitters releasing, neurons releasing neurotransmitters, gland cells releasing hormones. Those are all kind of neuron big things. There aren't glial cells on this picture. I'm setting you up for the surprise. Okay, so back about 20 years ago, right about the time that Marion Diamond had discovered that Einstein's brain had a lot of glial cells, people started using these fluorescent molecules, like the one I've shown here, that bind to calcium, and then they change their fluorescence. They basically get brighter. So fluorescence is where you shine light, like you shine UV light on something and then it emits light, like a blue light or a red light or a green light. So it gets excited and then it releases the light. So you have this phenomenon of fluorescence. And chemists have created these really clever chemicals that will do that, but the amount of fluorescence can be an indicator of how much calcium is there. And so when that technique was developed, neuroscientists thought this is excellent because, and a lot of people thought this is great because now we can, in real time, in living cells, we can see what's happening to calcium. And we can basically study things like muscle contraction, release of neurotransmitters at nerve terminals. Okay, makes sense. You've got a device, everyone has now fluorescent microscopes in their labs, so you can just set these up, you can load in these calcium indicators, and then you can watch what happens and see what happens to the calcium signal. Okay, so people were doing this, having a lot of fun measuring intercellular calcium, and we're learning some interesting, but not terribly earth-shattering things. I mean, they were learning that yes, indeed, calcium is required for all of these things that we already knew about. Before I tell you about the surprise, I just wanted to give you a little bit of background, tell you the work that I'm doing, because I'm going to show you the surprise as I discovered it, not as other people discovered it. So I work on the neuromuscular junction, and I have two pictures here. And so the neuromuscular junction is where a nerve synapses onto a muscle cell, and that happens at a chemical synapse. So the process by which that occurs is identical to the process by which nerve cells communicate with other nerve cells. It just happens to activate a muscle cell, and then the muscle cell contracts in response to that. And so what you have at the neuromuscular junction, so this thing right here, these three horizontal logs are muscle cells. Okay, they're going to contract this way. And the nerve that's coming from the spinal cord, this is the nerve axon, and it's wrapped by myelin. That's what this kind of outer fatty material is. And then notice what happens in this diagram is you get down to the surface of the muscle and the myelin stops, and then you have this little branch of the nerve. So the nerve then reaches the muscle and it kind of branches a little bit, and then from the ends of the branch it releases the neurotransmitter, which then gets sent to the muscle and causes the muscle to respond and contract. Okay. Notice what's missing here. This is from 1987. Miriam Salpeter, this is from a book of hers. There really aren't any glial cells here. Other than she's showing the glial wrapping up here, and she's showing a little bit of a terminal glial cell right here, but for the most part, the way this is depicted is of the nerve axon without any surrounding glial cells. And here's what it really looks like. And can you see that? Somewhat okay? So here's a picture I took a while ago, and what I did here is I labeled the nerve axon with a red dye. So you can see here's the axon coming down, and it sends off a little branch that comes over here, and this is where it branches over the muscle cell, and you can't see the muscle cell in this picture. Okay, you can just see the nerve. And these green cells are glial cells. And they're not very distinct in this picture, at least from my angle, but there are about anywhere from 7 to 8 of these things that cluster right on top of the neuromuscular junctions. These are actually Schwann cells, but they're non-myelinating Schwann cells. They're also sometimes called perisynaptic Schwann cells. And they cluster right around, if you look at these really closely, you'll see that these cells are sitting on top of the neurons, and they're kind of wrapping around like they're hugging these neurons, and they actually send processes that run all the way around these nerve endings and come into really close proximity to these nerve endings, which are releasing neurotransmitter. The same thing is true in the central nervous system. I just am not showing you that. So the central nervous system has a really similar arrangement where you have, every time you find synapses, there's a cluster of glial cells that surround those synapses. So here's the surprise, and this was done, I didn't do it first, this was done first by a guy named Phil Hayden, who at the time was at Iowa State, just down the road. He's now since moved to Tufts University. But he was one of the first people to do this experiment, but I'm not going to show you his stuff, I'm going to show you my stuff. All right, so what you're seeing here, what I'm going to show you, is an image of taking that calcium indicator, loading it into these glial cells at the neuromuscular junction. And when the calcium level goes up, these will get brighter. And so the way I did this experiment, let me see if it works. Okay, there we go. My experiment worked, yeah. That's how I felt at the time. Okay, so notice that when we start, it's dim. There's not a whole lot of light there at all. What I did is I stimulated the nerve. So I didn't stimulate the glial cells directly. I electrically stimulated the nerve, and actually I gave it about a fairly rapid stimulus for several seconds. When you stimulate the nerve at a synapse, notice what happens. I'll show it to you again. The thing lights up. It's the calcium went up inside the glial cell. Here's the same thing, but this is with pseudocolor, and pseudocolor is a way of representing intensity by converting it to a color. It's like what the weatherman does, where, purple is not very bad, and then orange and red gets more and more fierce. Same thing here, so just you can see the same experiment, just a little prettier, okay?
Needs music.
What?
Needs music.
I don't have music. Maybe Eric can help me with that. This is speeded up, by the way. I should mention it. This is actually taking place over several minutes, and I've sped this process up. Here's one of my favorite experiments. This is showing now a lower power, the same thing, and you'll see that it'll pop out at you several synapses. So now I stimulated the nerve, and I just watched what happened to these glial cells that are, oops, didn't want to do that. Watch this. I can play some music. So I'm doing this multiple times. And notice the prep moves, and that's because this particular muscle, when I stimulated it, I couldn't prevent it from moving. But I did this about 3 or 4 times, and each time you see what happens is they light up, and then they fade down really slowly. They light up and they fade down really slowly. And it's still such an amazing sight to see after all these years, but the first people who observed this just couldn't believe it. I mean, this was analogous to, imagine that you were studying a company, and you were really interested in some Fortune 500 company, and you were writing a book about it. And so you had been carefully studying all of the emails that had been sent between the executives of the company, their board meetings. You basically were following how all this information was being moved around in order to produce this product. And you were quite good at that. You got really good at decoding these email messages and figuring out how this company worked. And then all of a sudden you found out that the cafeteria workers, the janitors, the daycare workers, what was the other one? That they were all actually also taking notes. They had another way of communicating with each other. It wasn't the way that the executives were communicating through their email, but they were also noticing what was going on, and they were sharing notes with each other, and then passing that information on and controlling the company from underneath. So it was kind of like, imagine the analogy of a revolution, where suddenly you realize that, oh my God, these cells that we thought were just sitting there doing their jobs are actually aware when the neurons around them at synapses are becoming active, and they're generating these signals. And so I'm going to stop there, because Joanne is giving me that look. I didn't finish, we're going to finish it out, so what I'll do next week is I'll finish this lecture, and I'll talk to you about what we've then learned since. And so that was really the beginning, Marion Diamond's work, the discovery using this technique that glial cells are not just sitting there, but they're actually becoming really excited. They're doing something very interesting that this technique allowed us to see. And then I'm going to tell you a little bit about what we've learned in the ensuing years about what these glial cells are doing.
Thank you very much, Clark. Can't you just feel all your little synapses warming up? I hate to be a spoil sport and end the class this morning, but the one thing I got from this class this morning is that we should eat a lot of chocolate cake.
The sadder the better.
Judy Hunter, a very treasured member of our planning committee, has asked to make an announcement this morning. Judy.
You may remember at the end of the session last fall that Joanne introduced all the members of the planning committee. And our planning committee was talking, and we thought that perhaps the most important part of the committee, it was not recognized in that introduction. So we would like, as the members of the planning committee, we would like to thank the one person whose organizational abilities and leadership and care make these bucket courses possible. Joanne? A token of
Thank you very much. First of all, it makes me wonder what else is going on in the planning committee that I don't know about. You all know that one person doesn't make anything happen. And we have the most outstanding members of a planning committee that any chair could ever hope to have. And they are the reason this all happens. I may have had the germ of the idea, but without them, we wouldn't be here this morning. So in return, I thank the planning committee for doing just an absolutely marvelous job and for the success that we've had. And thank you, planning committee, for the recognition. I appreciate it very much. And thank all of you for coming, and we hope to see you again, same time, same place, next week.